Can everybody see the screen okay? Um, just somebody speak. You can hear my voice, right?
Yes, we can hear you and we
Okay.
can see you.
Okay. And when I change this slide, did it change?
Yep.
Okay, slides are changing. Good, good, good.
It's
I
a little delayed, but it's
It's
okay.
okay. Well, so here's the extra complication. Somebody can't make it right now for a good reason, and I'm trying, in addition to everything else, to record at least the slides and what I'm saying right now. So we got to see how this all works. I may have to abandon that part, but this is really trying to make everything work. We will get started in 10 seconds. Well, so I want to make a comment as we get started here about COVID and my behavior and what we're doing in the class and what I think. And I can't speak for anybody else, and it's not real clear to me what the college policy is on things. I think they're working more individually with people to be fair, and that's very nice of them. But Dr. Scott Gottlieb is the former head of the Food and Drug Administration, had a lot to do with important policies. And every Sunday he's interviewed on Face the Nation, and he always provides insight for me that sort of I can read the articles and things, but I need somebody to kind of put it in perspective. Right now, there's a standard for sort of thinking that we're in good shape for COVID, and that standard is, now you got to kind of listen, 10, 10 new COVID cases for every 100,000 people. So no matter where you live, that's just a scale, right? So big towns, little towns. So for Macoupin County, we have only 46,000 people. And what that would mean then is for us, the standard would be 5 new cases every day for our population in Macoupin County. And we're currently at an average. Macoupin County's got trouble because they haven't been recording for a lot of days, but it's at least 50. So we are at least 10 times the standard of letting up anything, and it gets worse. As Dr. Scott Gottlieb explained on Sunday, that the CDC standard is not five for us, not 5 new cases per day. It's 5 new cases per week, which makes us out of whack by 70 times. So I'm being really, really cautious. But there's hope. And so as I look at the data, clearly cases are coming down. So you went from screaming really bad now to only bad. And it's still real bad, but it's getting better. And so I have a lot of hope. And my hope is that when we come back from spring break, maybe things can return to pretty much normal and we always meet in class. So we'll look forward to that. But meanwhile, we're going to keep working like this. And thank you for staying in there and bearing with me. Right now, this should be the last lecture. I've got some things I want to wrap up, some big points to make about process of science and a little bit of the philosophy. Again, steps of the method is not what we're doing. I want to talk about big things, and there's sometimes I think about this, and I think it's the stuff that is in the cracks, the things that people don't talk about. But when you're a scientist, you expect people to know. So maybe we should spend some time on that. All right. In any case, there's the reminder of the test this Friday. Morgan will be running the test. She has a copy of the test and is ready to go right now. We hope to get it graded and grades back to you real soon. Is Tatum here? Tatum, speaking up. Tatum, paging Tatum. Earth to Tatum.
Yes, I'm here.
There you go. I got those quizzes ready for you. I didn't forget about you. You wanted to
All right, good deal.
Yeah, you wanted to review some quizzes. So I mentioned this and tell everybody about what Tatum wanted to do, which is review some quizzes, because that's what we have available for all of you. We want you to come in and see what you've done on your past quizzes and where you need to improve. Okay, so that's really, really important to learn from your mistakes. This is, as you can see from the lab quizzes, every week. It doesn't let up. We don't take a lot of breaks. Got to be on a schedule. And I hope this is the goal, that you are developing new study habits that allow you to get on top of things. So just like training for a sport, Of course, as you get going, it's a little rough, but we hope to develop new habits for you. In any case, reviewing your old quizzes is going to be really helpful so you know exactly what you missed, and you know that you can come into any study session and see that. I brought in, of course, yesterday's quiz that's ready for you to review today or tonight in any of the study sessions. Morgan, are we going to be okay staffing, or do we need to make any announcements on study sessions?
No, Cheyenne and I were able to cover all of them. So we have the normal 6 to 10 for this week. There may be a change on Sunday, the 13th, but we're still working on that depending on what we hear back.
Okay.
So
Jasmine's out for a little bit, and thank you, Cheyenne and Morgan. And I know if there's any changes, you'll email everybody right away. So that's why you always have to check emails every day. All right, let's finish this up and talk about some big points. We were talking about lots of things about the process of science. One is that we love repeatability. We love the fact that when we get one result, one of the very first things we do to test it is to repeat it. And I'm going to talk about that at the end of today's lecture. We also said we like large sample sizes, and we gave an example of pulling cards of different colors. And I told you that in a small sample size, if I'm got a deck of cards, we'd expect there's a 1 in 4 chance to pull any one of the suits. So if I wanted to pull a heart out of a deck of cards, it's a 1 in 4 chance. But if I pull 4 cards, it may not be a 1 in 4 chance. It could be that I get a couple hearts. But in a large sample size, if I pulled 1,000 cards, right, pulled one card, put it back, did that 1,000 times, we'd expect this to be pretty close to the 25%. So we said you could feel a little lucky by the bias of a small sample size. And I just said something pretty big. It'd be nice to write down. What I largely think that people are referring to when they say they're lucky or unlucky is the fact that something unlikely is happening. Maybe if they're going in and gambling or playing a sport, it's the bias of a small sample size. That if you went and played cards or something and you played 3 times, that might turn out to be all against you or all for you. But if you keep playing, we'd expect things to even out. That's what we're saying with large sample sizes. So you got to be really careful when you go into the casino, and that's what I was telling you. You got to know when to hold them and know when to fold them, as Kenny Rogers sang. A billion years ago. Your parents may not know that song. We talk about margins of error. So when we get results, it's important to understand that. Now I'm recapping here. When you see an average, what we care about more in science is a range. And that means it's the average plus or minus the margin of error. So if the margin of error is plus or minus 3 and the average is 40, the range would be 37 to 43. And Dr. Bray, just to give him a compliment, has been one who's always been pushing this when we say data from the college. People often report an average, and we want a range. We care a lot about the range. And that has to do, remember, with sample size and how you selected the data. So we said that if your sample size is really large, we'd expect the margin of error to be smaller. And that's something you might want to write down. So margin of error is larger when sample size is smaller. Because again, a small sample size is not as representative. We'd also talked a couple days ago about what builds confidence. So these are really big points, guys. These are things that I don't think we've talked a lot about in grade school and high school. What builds confidence are many good reasons. That's essentially what we're saying. Multiple independent lines of evidence. Okay? So they're independent, they're separate lines of evidence, like fingerprints, for example, and videotape of license plates. Those two things are not related. Okay? They're independent. And so we like lots of lines of evidence, and I used an analogy of a cell phone tower or some tower held up by guide wires. And finally, and I boldfaced this because I don't think I talked about it before, but I've mentioned this many years when I teach zoology. As we go forward, it's very likely, I hope, that you bring up things that you've seen or heard. And it might be some crazy things that you've seen or heard. When I say crazy, I mean, wow, it's just really unusual. Okay? And the normal thing that we should do in science, remember, is fold our arms and resist that idea. We want to know more about that. When we are told something, we do not just accept that it's true. And that's called critical thinking. And that, oh my goodness, is so important right now when you're going to wherever you're getting your social media, whatever it is. I've been on the internet before. I watched somebody who was a weatherman, supposedly, who was out by traffic, and it looked like it was a weatherman, a young guy, who was just complaining about how stupid it was that he was doing a traffic report. And then I looked into it and found out it was just a comedian who set it up. But it was being forwarded as if it were true. Okay? We now know that we can fake video. And this is becoming worse every day as people get better. But if you haven't seen the Tom Cruise faked videos, they're really, really good. And I've watched programs on how the guy did it, how they actually put it together. So, when we see things that are really inconsistent with the world we know, okay, we should doubt. So, one of the things about building up our confidence is, if you found out that a rock was hard, somebody went out and got a rock and it was hard. So, here's a really cool rock that is in my office, and it's actually a petrified sand dune. Not sure that you guys can see this. I'm trying to get the good light on it. And I don't think you're surprised to find out that this is hard. Yeah, okay. So that's consistent with the world I know. And, that's good. I'll believe that. That seems likely. But if you told me that a bunny, like a little bunny rabbit, ate a dog, first thing I do is fold my arms and go, are you talking about a real dog? Or are you talking about like a stuffed dog? And then if you tell me, I'm going to ask questions. And if you tell me, no, I mean a real dog, I'm going to doubt it. Okay? So in science, it is good that we doubt. We doubt things that are inconsistent with the world we know. And one such example was a discovery about 30 years ago that some ulcers, stomach ulcers, are not caused by stress or spicy food or something. They're actually caused by a bacterium. And the evidence for that had to be overwhelming for people to believe it because it was so unique. And it's really, really cool. And if you take microbiology, you'll probably learn about that. Okay? And you should know that those people got the Nobel Prize in medicine. So how about that? Something that we had no idea was true, that modern medicine said, yeah, I don't think so. And so we can be convinced with lots of multiple independent lines of evidence. Okay. All right, we're just moving on. Now for some test questions, some things just to review some of other aspects of science as we go on and finish this up today. So go ahead and read this one and see if you can see what you put down. And this is a problem that you will face when you're researching things on the internet. When you go to a web page, is the information there simply what someone thinks or, their experiences? or are they citing scientific research? Can someone help me to understand what peer review is? What's peer review? That seemed to be important here. Give it a shot.
Well, what peer review is, you have like one of your colleagues like look over the study that you did or what you conducted, and just to make sure the data is consistent.
That it makes sense. So it's like a good harsh look, right? It's a good feedback. It's more formal than that, Logan. So you're on track, absolutely on track. I want to describe for you peer review. I have been a peer reviewer. Right now in my office, I'm talking so loudly that Dr. Luth and Dr. Welch can probably hear me right now. So Dr. Welch might hear me actually I wonder if he's been a peer reviewer. Here's the thing about peer review, Logan. It's much more strict than that. So I've published, about a dozen scientific papers on all kinds of different topics, and that is peer-reviewed scientific articles, okay? In some pretty top journals, to be honest. Journal of Parasitology, Journal of Morphology, American Society of Ichthyologists and Herpetologists, other kinds of things. When I submitted the paper, I submit it to the editor of a journal. And this is usually a print journal, but now we're going to some online, but still it should be peer reviewed. Okay, here's the process. So you want to diagram this out for yourself in your notes. A scientist, like you, submits a paper to a journal. And there are guidelines in the journals on how to do this. There's formatting issues. There's parts of a paper. Some papers have an introduction, a materials methods, a result, and a conclusion, and an abstract and a title, of course, and a lit site. And some papers kind of smear that all together. And some of the biggest journals, Science and Nature, don't have those distinct sections. So you have to follow guidelines and you have to format the paper for the appropriate journal. Then when the editor receives it, so you got to listen carefully, this is important, that editor knows other scientists working in the field, usually 3, but it could be up to 5, and that editor contacts those people, as I was contacted, and an editor says to me in an email, hey, Dr. Zelisko, I've got a paper on this subject, and you seem like you're an expert in this field. Would you mind reviewing it for us? And sometimes I've been too busy, and most of the time I've said yes. And I say yes right away because it's professional. And then they send it to me. Now, Logan, here's the key part. The person who wrote the paper never finds out who I am. It's anonymous. Now, why do you think that it's important to be anonymous as a reviewer? Jesse, what do you think? How does it help to be anonymous?
In case you say something that they don't like and they get mad at you or something.
Yeah. So you think I'm more likely to be honest?
Mm-hmm.
Yeah. Hey, let me apply this right away to your life. When you ask for a letter of reference, so I got some important things to tell you. You might ask me for a letter of reference sometime. So please make a little note over here about letter of reference. So let's go. Right outside my office door here, literally 8 feet away from me right now, is a folder on a little desk outside my office. And I have written directions on how to ask for a letter of reference. Morgan remembered that I offered such a thing a few years back, and she needed a letter of reference from me and other people. And it's a really good reminder for students on when you ask, honestly, anybody for a letter of reference, there are things that you just, you're new to the process probably, and things you need to do. So I'd recommend that all of you come outside my office when you can and grab one of these advices for letter of reference. Well, related to that, when I write a letter of reference formally for you, when you request it, you have to sign something that says, I waive my right to see the letter of reference, or you don't waive your right. And before I submit the letter of reference, I see whether or not you waived that right. Back to what Jessica said, if you waive the right, then what I, these are rights, it's using two different words. W-R-I-T-E, what I put in the letter of reference is then anonymous, right? To you. You don't know. You're not going to be angry with me. And you should know that if you ever request a letter of reference and you don't waive the right, you should expect that the importance of that letter will be discounted. That is, people won't put so much weight on the letter if they know that you wanted to see what I said. So it's the exact same thing for peer review. If I'm going to stay anonymous, I'm going to write more of what I think. And sadly, two of the papers I reviewed, I was really hard on the people. I don't want to detail what they did wrong. I'll say this. One guy wrote a paper. It's a person that I could go drive to and see tomorrow. He works in my field. And he completely ignored lots of research. He had no idea. He was acting like he was the first person to do something. And it had been well worked out a long time ago. And his paper was meaningless because it had already been done and he didn't even know it. And so I was rather merciless with him. Another person used some techniques that screwed things up. I'll tell you this much because I don't want to get anybody in trouble. It's been published. They were measuring the speed at which salamanders move at night, and they were using flash photography. So they were taking a photograph of a salamander in one place, and then a few seconds later taking a photo in another place. And they were saying this is how fast they normally move. And I said, do you think a flash photograph might scare a salamander? In any case, that was one of my concerns. But I was hard on the people because I'm part of the scientific process. The reviewer really points out important mistakes or concerns that you might have. Okay? So peer review is so important. And that's why in an awful lot of COVID reports, we are anxious, as we were, to find out if Omicron coming out of South Africa was less severe for causing disease and if it was spread faster. And so regularly, if you listen to the news, reporters would say, well, the first data coming out, and it wasn't peer-reviewed yet, they'll say. And so you should know, okay, well, there could be problems with the study, so we got to be careful. So they regularly, guys, you're living in a time of so much science and the process of science. So the fact that an awful lot of this data hasn't been peer-reviewed, well, the problem is this takes months. And you and I want some idea of, look, is there a new variant that I need to be worried about? And we really hope that by the summer, guys, Omicron's faded and we're in really good shape. So I'm very optimistic about the summer, but I'm not optimistic about next fall. I think there could be a new variant and we're back in the same thing again. So peer review takes time. And in a fast-moving COVID world, people have been reporting things that haven't been peer-reviewed. But again, peer review is anonymous. So regularly as scientists, before we trust any science, we ask the question, yeah, but has this been peer-reviewed? And maybe it has and maybe it hasn't. Why hasn't this lighting on me? I've got a lamp right here in my face, and it's not making me light up. I think it's important to see your professor. Let's try that. All right. So peer-reviewed matter. There might also be things that I put on my website that just said, yeah, this is what I saw. This is what I think. And that's okay. But you should know that's not something you're going to cite or put in a paper. The fact that somebody just thinks something. Okay. You want to see what their research is. You did really well on this question, so we don't have to spend a lot of time. But when we try to check to see if some scientific report, especially if it's really important, and you know what? I'm just going to tell you the story now instead of later. In spring of 1988, how about that for being specific? In the spring of 1988, I was teaching a general biology class, and I walked into the class and I said, guys, I really need your attention. You really want to listen up. Something was reported today, which if it was true, spring of 1988, you will have essentially free and unlimited power, like electricity. Free, nearly free, and unlimited. Because they reported cold fusion. Now, fusion is this really powerful reaction that can be used to generate electricity. And if you could get it happening at room temperature, oh my goodness, guys, it would transform our society. Just imagine that you'd have all this electricity and it's not a limiting factor anymore for us. And it was by two scientists, Pons and Fleischmann. And now here we are, what are we looking at? 34 years later, people immediately tried to test it and say, okay, well, let's do what they reported and see if we can get it to work. And we could not. Pons and Fleischmann have been interviewed on 60 Minutes and other places, and they want you to know that their university was sort of rushing them to report something, that they actually wanted to take more time. And there actually may be a little bit to what they reported, but obviously it's not transformed our society, so we're not there yet. But we do hope to use fusion reactions to generate electricity. The point was a major, gigantic, earth-shaking, humankind would change report came out, and immediately physics labs all over said, well, we're going to try this too. And they couldn't. And I can't think of a better example of repeating experiments. Moving on. Talking about the process of science, and one of the things I want you to know is bias. So I have a quote on my old office door, still there, I think. And the quote is this. It's not from me, by Sir Peter Medwar, is actually who it was from. And the quote is, innocent, unbiased observation. Innocent, unbiased observation is a myth. That each of us, if we go out in the wild and look at the world, bring with us biases. And as a zoologist, I want to tell you, and it's something I've really grown at in my thinking and teaching the class, is I've become so much more aware of how limited our perceptions are of our world. So you can't hear really low notes and you can't hear really high notes, right? You can't hear bats. Bats can hear high notes. You can't hear low notes like elephants use to communicate. You can't see ultraviolet light. A lot of insects can, and we think a lot of birds can, interestingly. You can't see infrared. Does anybody know who can see infrared, which is heat? It's an animal a lot of people are frightened of, and they exist right here in our county.
Can I answer?
Yeah, anybody.
A snake?
Yeah, snakes. And particularly, it's probably rattlesnakes. They have heat pits in their face. And my mentor, Dr. Ken Cardong, who's no longer with us, Dr. Cardong studied those heat pits, and he found that the nerves from those heat pits went to the same part of the brain as vision. And he actually hypothesized that when they see something like you and I do, that the heat image is superimposed over the visual image of light that you and I see. So we don't see the world the way it is. We see it based on our own senses. Further, when we interpret things, we interpret them based on our experiences. So what I'm telling you is, guys, bias isn't a mean thing. It isn't because you're evil. It's a recognition that we can't see everything innocently. We bring with it personal biases. When people observe animal behavior, they often think things are cute and cuddly and that they're loving. Well, that may not be it at all. In fact, animal behavior is really a difficult field because you have to interpret the behavior from the perspective of the organism, not from you and I perspective. And our perspective, adding human traits to other non-human animal behavior, is called anthropomorphism. It's where we project our human traits on other critters. And yes, it's really fun to watch animal films and do that. And that's one of my favorite pastimes, is actually to put voices on little animals. I like to do that. I think it's amusing. And some people like to watch me do that. But it's not real, right? So we are all biased. And one way that we try to reduce biases is use equipment, recording, measurements, right? So we try to have really good thermometers. So we don't go outside today, and hopefully it's in the 40s, and say, yeah, it feels pretty good. We use a thermometer, okay? And we measure things. We measure sound at levels that we can't perceive, light at levels we can't perceive. And we try to reduce our biases. Guys, this is so important for zoology, the class I'm teaching and telling you right now. We are hugely biased in our senses and experience in zoology, and we have to work so hard to avoid it. Now, sometimes biases are deliberate, or the fact that you're optimistic, okay? So a scientist might come up and think that they've got a drug Z that will literally overnight stop all cases of COVID. Is that going to be the best person to go out there and give the drug and assess the results? No. That person, when they give that drug to people, hoping that people get better, that person might sort of see the positive even when there's not positive and report it because they're hopeful. Drug approval is a very slow process. And it tries to remove biases by doing things like this. By conducting, I got the rock I showed you was sandstone. I have sand all over my computer right now. Oops. One way that we reduce bias in scientific experiments are double-blind studies, and only about 40% of you got this right. So let's make sure you know what that means. My wife and I, struggling with my wife having Alzheimer's. This is devastating for us, but nonetheless, we joined a scientific study that might give us a little bit of hope in St. Louis, and it's a double-blind study. Half of the people who are in this study, and we just finished 2 years in this, half the people are not getting a drug, and half the people are. We don't know who is, and the people who are injecting my wife don't know either. That's the double blind. Those who are in the experiment and those conducting the experiment. Now, someone else knows, right, and collects the data, but the people involved, both the patient and the practitioner, the person who's administering the medicines, they don't know. If you're studying bee behavior and you're going to do an experiment with bees, are you going to do a double-blind study? Logan?
I'm thinking about it.
Yeah.
I don't think so.
Why? What's different than the study my wife's in?
Well, yeah, I mean, it doesn't necessarily, like, you're not studying humans. I don't.
Right. Well, so how are we different than bees? When we study humans, how's that different than studying bees?
Well, we're studying ourselves at that point if we're studying humans, and then if we're studying bees, we're studying like arthropods.
So how does that affect the experimental design? Anybody want to help out?
Oh, we're not biased.
Well, the bees aren't going to really be talking to each other and saying, "Hey, Phil, did it look like you got water with sugar in it? Because I ain't tasting any sugar in the water," if that's what you were testing, right? Right now, I'll tell you, in our experiment, just being real life, so this is pretty cool. In our experiment, about half the time when you get injected with the drug, you have some kind of reaction. It might be a rash, or in some of the tests like MRIs, they have different problems that show up. It doesn't always happen, but my wife has never had any reaction to the injection, and all the medical tests they're doing, everything's normal. So we think we're in the control group. Bees are not going to do that. Yeah. So when you're a zoologist, unless maybe they're really intelligent animals, like perhaps great apes or elephants or dolphins, they may somehow, I don't know, I'm being really cautious here, you might possibly need to do a better way of disguising the test and the control groups. But if you're injecting a drug or something like that, you don't tell a dolphin you're getting the medicine and the dolphin next to you is not, right? So double blind is mostly for human studies. And the reason is that, of course, if you think you're getting the drug and you think it helps, then you might start reporting positive things that aren't true just because you're hopeful. And there's lots of variation in behavior. Amy has some very good days. Last Tuesday, I called her up on this really long day. I have 6 hours of lab today and 1 hour of lecture. And I called her up last Tuesday, and she seemed like her good old self. And I just called her up before lecture, and she seemed like her good old self. But sometimes in the parts of the days, she's a little more lost. And so there's lots of variation. So are we going to emphasize the positive or the negative? See? And if we're getting the drug, we're going to emphasize the positive because we want that to be the case. Moving on. This is now at the very, very end. This is big stuff here. We talked about how science is self-correcting. One was we said we repeat, right? We tend to repeat. And already right here on the cold fusion, that was the story I already told you, trying to repeat. I want to be clear. The scientists, as I remember their names, Pons and Fleischmann, are not bad people. They did an experiment. I think they're still unclear about what they did that got the results they did. And I still think they're trying to work things out. But whatever they did, by mistake, they got a result. And other people couldn't repeat it. So I want to make clear that the scientists make mistakes. We don't sometimes know why we got a result. We report it, even in a peer review, and we just can't repeat it later on. So we have to go back and really look. But people who deliberately cheat the system eventually get caught because no one can repeat what they're doing. But number 2 here, the failure to build, that second one is just as important. So I need you to know this. In an awful lot of research, people don't just go out and repeat the experiment. What they do is the logic of if-then. Well, if that's true, then this should be true, and I'll go do that experiment. And in an awful lot of cell biology and molecular biology, we build on other people's results. It's not that we repeat it, we extend it. That might be another one of those good words. I tell you that when I lecture, I try to tell you maybe an extra different way of looking at it. So if it's wrong or false, and again, it could be cheating or it could be just mistakes, and somebody reports this and you go, wow, if that's true, then this should be true. Okay? And if we can't build on those prior results, then there's a problem. Then we go back. And this is important, the next point, for COVID, because I was hearing some of this come up in our town halls about COVID. There is no strong personality that wins in science. So why do we think falsely that something's true? Well, because this hotshot keeps saying it. Yeah, well, that hotshot needs to have evidence. Okay? And having a scientific conspiracy. So I'm about to tell you something that's really big, because this is largely the recap here. I call this, and this is just me, I call it my young punk strength of science. Young punk. And I mean that nicely. What I mean is fresh PhDs or grad students come into the world of science. They learn about all kinds of things people know. And they would like nothing better than to bring down some big idea and make their own name. And there is fame and fortune in science. You can get really well paid and have a really great lab if you're doing important work. My point is, if any hotshot person has ideas that aren't really true or need to be reworked, there are always, and that's why I mean this in a fun way, please, the young punk new PhDs are going to retest those ideas. They're going to try to chip away. And by the way, when they can't, that increases our confidence. But sometimes young people come along and they think about something from a different perspective and they really add to important ideas. And I want you to know that Darwin and Wallace, we're about to talk about, made their greatest contributions in their 20s. I'll give Darwin his young 30s. Albert Einstein, anybody know about when he did most of his important work? In his 20s. For you folks going out into science, you get your basic broad education, and now in your 20s, you start learning a lot, and I hope you question. And it seems to be an important time in your life after you graduate that you really start learning enough that you really start challenging all kinds of ideas. Or you start realizing the potential of a new technology because you're young and you used it, right? So for example, and I mentioned this before, people going into public health who use Google a lot probably suggested the idea, which is true, and they use it, that the CDC looks at where in the United States people are Googling things like flu symptoms and flu treatments. And that correlates with a flu outbreak. So where you're doing a Google search with certain kinds of disease gives us an idea that that's where there's more disease. And that's probably because people were really hotshots with the internet and then they entered the field of public health. Who knows what technology you'll think of next, but we're going to have all sorts of cool technologies. So the metaverse. So Facebook's now, the company's called Meta, right? Yes, no? Am I not right? Yeah. So what is the metaverse? And why is a 64-year-old man explaining technology to 19-year-olds? What's the metaverse, guys? No! A 64-year-old man cannot be explaining to you something that's huge in technology. It's a make-believe world. And you could have goggles on, and you could enter this metaverse, and you can have your own personality, and you can buy real estate. People are currently selling real estate that, like, they could sell my house, an image of my house, but in the metaverse, and then you can buy it, and there will be a currency exchange. So it's a fictional world that we sort of all join, and you can be who you want to be, and you're going to behave and interact with people and have stuff in the metaverse, and there's people that think you're going to buy that. Just like you can buy extra game tokens and things for playing apps on games, you can pay to have an advantage, right? In any case, who knows where that will help with medicine? PTSD. So people are trying to get through and past horrible things. And maybe they can be in a metaverse that helps them somehow as therapy goes. Okay? So creative thinking, guys, that's what I'm saying. It's new young people coming along right here that are going to pull down any bad ideas. So I want you to know there's no conspiracy. Dr. Anthony Fauci, if he ceased to be around, all these good ideas he has are either good ideas or bad ideas. They're not good ideas or bad ideas because Fauci said them. They're good ideas or bad ideas, okay? Either masks are good or masks are not good. It doesn't matter if Fauci says them. And yet his life has been threatened. If you didn't know, there are horrible threats against him and his family. So it's not about a conspiracy in science. It can't work that way. People will bring down that. All right, we talked about bias already, so we're pretty much through that. Talked about peer review, helping things on the internet. That was that question. This is the last slide. So what I want to end is, not only as a person entering science should you understand the process of science, but you should understand that science impacts our society in great ways. I didn't even list the space program here. A little trivia for you guys. My exact age of life is within a month of the age of NASA. NASA was officially created 1 month before I was born. And it was created, does many know why we suddenly had a big space push in winter of December of 1957? Do many know what happened in 57? Russians put up a satellite. And we were losing the space race. And so the federal government, big time, jumped in and created the NASA and looked at going up into space and putting manned people in space. Right? But what else has happened? Endangered Species Act has happened, and variously, depending on which president. So when Trump was president, they were reducing protections for species. And now that Biden's president, we're protecting species more. But you should know the history of doing good things in science is not restricted to parties. So does anybody know who was president in 1970 and 1972? I really want you to know.
Richard Nixon.
Absolutely. And so those two wonderful things, Clean Air Act, Clean Water Act, were passed under President Nixon. Right? Democrats and Republicans came together, and they can come together to do good things. If we can get past our team approach and start looking at what's best for all of us as policy. So these are really, really important things that have made and cleaned up the air and water. Right now, laws, all kinds of laws are based upon science and what about it? So stem cells, President Bush, the second Bush, came on and had a national televised address to the nation about stem cells. And I was teaching cell biology at the time. So can you imagine how exciting it was to be a cell biology teacher and the President of the United States comes and addresses the whole nation about stem cells? Some stem cells come from aborted fetuses. The abortion happened either way. And I've struggled and asked, why would anybody object if an abortion occurs? By the way, it could be a natural abortion. So now we have a fetal body, a body of a fetus, and we could bury it, or we can incinerate it, just like people get cremated, or we can take some of those cells and try to help people. And I wondered, why would you not want to try to help people? And one of the answers, I don't know all of them, is that some people so much object to abortion they don't want anything good to happen from one. And I was like, ugh. Okay, I don't want people to be shot and killed. But if I'm shot and killed, you can use my cells for research. That'd be okay. So anyway, President Bush, again, was talking about how they could be used. Human cloning. So if you really like yourself so much and you got a lot of money, can we just make another one of you? And you should know we have the capacity to clone you. We're cloning dogs. We're cloning cats. And if you think laws are going to prevent this, well, you can have enough money to go out in the middle of the ocean and do it out in the ocean, or do it in secret. So there are laws on human cloning, and we have to be very, very aware of these kind of things. Human genetic engineering. So each of us, right now, WashU in St. Louis is one of the best places. They are genetically engineering cells and changing things and treating disease. What we have banned worldwide is to change genetics of an individual in such a way that those changes get passed along when you reproduce. Okay, there's more complicated ways to tell you this, but that's what it means. We are doing genetic engineering, but not in cells that are involved in reproduction. But right now, you can do in vitro fertilization and have a little 4-cell embryo, and you can go in and change the genetics of all those cells, and when that little embryo grows up and reproduces, it will pass along those changes. That's illegal. Now, that's for your future generation to decide. And finally, GMOs, which you may or may not know, genetically modified organisms, which mostly have been plants, but we have some salmon that are genetically modified that grow twice as fast. There have been various countries with restrictions. This year, the labeling of GMO products changed, and it's not as restrictive. We have regularly had plants, as Dr. Bray must have told you, that are genetically modified. And that was done by just crossbreeding and all kinds of selection that we've been doing as we've created artificial strains, cultures of different kinds of plants. We did that more or less haphazardly and randomly in the past by just crossing things and seeing what emerges and then raising those, like you might do in developing a dog breed. Now we have the ability to do it very specifically, go in and change a specific gene or a specific allele. And it's much more targeted and sort of clean in that way. And there are potential concerns, of course. What are the properties of anything that's genetically modified? But there's also a lot of safety tests. And I don't have concerns about me eating plants that have been genetically modified. I do think, I guess, sometimes about the meats and being genetically modified, but that's where government regulation is coming in. All right, that to tell you, we're done. We're going to talk about evolution and pick that up on Wednesday. That's tomorrow. We'll do another Google Meet. Expect Google Meets for lecture for at least this week and next week. We're always assessing, and I just be exciting if COVID cases drop off a cliff. But this Friday, then, you have all the information you need. On your lecture outline for lecture exam 1, you have a study sheet, and that reminds you of what I think you should have gotten out of lecture. Okay? And that's the only time I do that on regular lecture exams, because the other ones are more straightforward and match the book. But this one, I think it's fair that students could say, what did he think I was supposed to get out of that? Okay. So I'll see some of you at 12:30. Of course, always the morning people, if you wanted to come in the afternoon, there's room in the afternoon for some more study. Morgan, somebody asked whether or not you might be able to dissect earthworms in study sessions. Yes. So you'll find a jar of undissected earthworms in trays, and if you can give them some help or guidance, but they might have more time to do that in the evening. So some people are doing it during lab time and others are doing it later. Okay. Questions on anything?
I kind of had a question. It was from earlier,
Go.
a little bit.
Go.
It was before. It was like during the lab. I wasn't sure where to put the earthworm afterwards, so I wanted to ask you about it. But lecture was coming up, so
If it's covered with water, we'll just leave it there for other people to see.
it is not.
I will add water.
All right.
Okay. Thank you for telling me, Logan.
Yeah,
We're
I
gonna
was gonna catch you real
Yeah,
quick,
yeah.
but it's like, shoot, I gotta get over here.
No, I understand completely. We're just fine. Thanks for telling me.
All right.
Okay. Boy, get going and studying for that test, guys. See
Have a
ya.
good one.
I'll see you in lab. Bye-bye. Morgan, see you.